Okay, thank you to stay here to wait for my talk. I updated my talk content because this is something really new. I just finished it 3 days ago, but I would like to share with you. These two talks actually are connected. But for this one, I'm trying to emphasize how to develop a statistical method to quantify the impact of genetically regulated gene expression on complex disease and traits. This is joint work with my PhD students, Informationn, and my collaborator, Jin, from Duke NUS. In case some of you guys may not be familiar with the background, so I will begin with my background and then present problem formulation and method. And in particular, I'm trying to show you many real data results. I would like to begin with this movie, Beautiful Mind, which described the mathematician John Nash, who suffered from schizophrenia. And the life is really hard, and we sorry to notice that the son of John Nash also suffered from the mental disorders. And why I mention this, because we know that schizophrenia is a highly heritable complex disease. The genetic contribution to schizophrenia is roughly about 70 to 80%, and also for other complex disease like human height, we know the variation for human height are also greatly contributed by genetics variation. For example, genetics contribute about 70% of the human height. We call this as the heritability, and it also for some common disease like autism and type 2 diabetes, genetics also play a very important role. All these numbers are estimated based on family study in old days. And now, roughly in 2002, genome-wide association studies started to look at single nucleotide polymorphism, try to associate those kind of genetic mutation with the complex trait. The basic design about GWAS is like this. You can think of a big matrix, and each row represents one individual, and each column here represents one genetic mutation. For a commercial genotype platform, typically now we have about 1 million to 2.5 million SNPs we can have. We just need to pay about 200 or 300 Hong Kong dollars. That's good enough. And here we can also collect some covariates for those individuals, just like age and sex. And then the basic idea is trying to associate with this kind of genetic mutation and the disease status by perform association study. The simplest one is try to do this by so-called contingency table and then perform chi-square. Basically, here we are trying to check for what given SNP, and we are trying to compare the genotype frequency for cases and controls, and then we calculate chi-square value and the p-value and so on and so forth. But for the real data analysis case, perhaps in GWAS, we need to control, for example, the population stratification, those kind of confounding factors, and then calculate those p-values. But here, that is not my topic today. Anyway, after we compute those p-values, we can see this kind of plot. We call it Manhattan plot because these kind of dots, just like the buildings on the Manhattan place, in Manhattan in New York. And the x-axis actually is the index for human genome, and the y-axis is for the negative log 10 p-values. And this is a Manhattan plot I taken for type 2 diabetes. And this is, for example, this FTO genes is well known for obesity. Well, after a few years of GWAS study, and people realized, okay, there is something unexpected. What is that? For those GWAS hits, like FTO and these genes, those kind of GWAS hits, most of them are not located in coding region. Actually, according to the science paper in 2012, roughly only about 5% of the GWAS hits are located in coding region, and more than 90% of those GWAS hits actually are in the non-coding region. And people start to think of, so why it is the case, and propose to say, okay, gene regulation plays an more important role for complex disease and treat. And then in this talk, I'm trying to present a method to quantify the impact of genetically regulated gene expression on complex treat. And then I'm starting to formulate the model and then present the computational method to solve this problem. And my formulation actually was motivated from heritability estimation. Actually, heritability is an old concept which originated from the father of modern statistics, Fisher. And exactly 100 years ago, 1918, right? Fisher has a very famous paper. He introduced the random effects. Basically, T is the phenotype observation, and X is for the genotype, and here epsilon is for the environmental effects, and also the beta was viewed as a random effects, and Fisher defines something like this: the ratio. Here the variance for the genetic component, and here is the variance for the phenotype value, and the ratio is defined for heritability. Basically, it means the variance of the phenotype, the proportion of the variance of the phenotype can be explained by genetic components. That part is called heritability. And this is the definition in about 100 years ago. And well, a question is, can we revisit this kind of concept by using modern genomic technology? And people start to look at this problem maybe in 2010. And now, based on the collecting the data, we can estimate the heritability using GWAS data instead of using simply pedigrees data. And we can compute heritability for a lot of different phenotypes. For example, autism here, the heritability is estimated to be very high, roughly 90%, and so on and so forth. And as motivated by this kind of thing, we are trying to quantify genetically regulated expression, the impact of genetic regulated expression on complex disease and trait. So in short, let me just simply call it like IGREX, okay? And here's the main idea. We want to quantify the proportion of phenotype variance that can be explained by genetically regulated gene expression. I call it GREX, okay? So what is the key idea? That is, the variation in DNA results the change of RNA, and the change further lead to the change of the phenotype. That is one way. Of course, there are another part, that is the DNA affects the phenotype through some alternative effects, which is not through the gene expression. So we try to model this part. That is, we try to partition the heritability into two parts. One part is due to GREX. Another part is due to the alternative effects. And but one challenge is that for GWAS data, we do not have gene expression data. measured for the same individuals. Well, then we have to use some kind of other information. Last year, there is a Nature paper talking about GTEx project. Basically, it released a big database for different tissues. In total, there were 44 tissues for a human body, and for each tissue, there are about several hundreds of individuals whose gene expression and genotype are simultaneously measured for this purpose. And basically, we are trying to combine two types of the data together to formulate our model. The first part of the data is from the GTEx part. We have the genotype data and we have the expression data for example, for different tissues. Here, let me just show it for one tissue. We have, for example, about 20,000 genes in one tissue. Okay, and this is another part for the GWAS data. We have the genotype data, but typically we don't have the expression data. And the sample size now for GWAS data, typically more than 10,000 of the individuals. And we have their phenotype data. And the question is how to connect these two parts of data to answer our question, to quantify so-called IGREX. And here is our thought. We try to build a model based on the GTEx data such that it can build a relationship between genotype and expression. And then for the give estimate model, then we put the new genotype data to the model, and the output will be the imputed gene expression data. And then we use the imputed gene expression data to explain the variance of the phenotype. And that is so-called the proportion of phenotype that can be explained by genetically regulated gene expression. Okay? And how can we do this? We typically do this based on the following statistical model. First, we use a very simple linear model to relate genotype and gene expression for gene G, and G from 1 to, for example, 2000. And then this T is the vector for the phenotype value, and the first part is due to the impact of GREX, genetically regulated expression, because this is the genotype, this is the effect size for the gene regulation effect size, and alpha is the coefficient such that the imputed gene expression, the combination can affect the phenotype. And this part is for the alternative effects, and we define that just similar to heritability, to the definition of heritability. And we define that the variance explained by this part, the GREX part, and the proportion of variance explained by the GREX part as the PVE. We use this quantity to quantify the overall effect of genetically regulated expression. Okay, and then we start to estimate those parameters, for example, the alpha and the beta. And how can we do this? Actually, from statistical point of view, there are many different methods, but before we do that, we should keep something in our mind, that is, the method should be computationally efficient and statistically accurate. So, based on these two considerations, we take two different approaches. The first one is given like this. We first give the statistical model some probabilistic structure. For example, the beta assign some random effects for beta, just like Fisher did. And also the alpha we assume is some kind of random effects, and the gamma is some kind of random effects. That's this kind of random effects structure. And then based on this kind of probabilistic structure, we can infer the posterior beta for the observed x and the y. That is the posterior for beta. The posterior mean and posterior variance can be exactly write down. And this is for stage 1. We want to have the estimate of beta. And for stage 2, instead of use the point estimate, we need this kind of posteriors, full posteriors. We use the posterior from the stage 1 as the prior for stage 2. And based on the so-called law of total expectation and total variance, we can have this following things. That is, given alpha, the expectation of the phenotype value can be written in this way, just like because of the model. Okay, just here, take expectation with respect to beta, then that will be alpha x and mu, because the gamma is assumed to be 0 mean, so that's this part. And then we further take expectation, then we know, okay, because alpha is assumed to be 0 mean, so we know, okay, the expectation of the phenotype vector should be 0. And so on and so forth. We can do this for covariance matrix. But anyway, we're using, again, we use the law of total variance. And we can have that the covariance for this phenotype vector will be given in the following ways. Here, I would like to emphasize one important thing, that is this part, the sigma g part, which is very, very important because it captures the uncertainty in the prediction part. Remember that we need to impute this kind of gene expression, but you know this is some kind of imputation. There exists a lot of uncertainty. So how can we quantify this uncertainty? That we should not simply use, say, like x times beta using the posterior mean to replace the beta. So that is not accurate enough because of the uncertainty. And we need to take care of those kind of this part. That is the posterior variance part. Without this part, actually, the later experiment is going to show you that this will result about 50% of the information loss. So we need to take care of this part by take the uncertainty into consideration. Anyway, with this, we have the covariance for t. And then we can according to the definition of this thing, the proportion of variance can be explained by graphs. We can plug in to have this kind of estimator to estimate the proportion. And the only thing we do not know is those kind of variance components. So here it comes two different methods. The first one is that because we know, okay, the expectation of t is 0, and the variance of t is this kind of matrix. So we can impose some kind of normality condition. Simply say, okay, t follows some normal, 0 mean, and the variance. Of course, this is expected to be not good enough, but it's very, very surprising. In the later on experiment, we show that, okay, this Remo method works extremely well with unbiased estimate and also very small standard deviation. That's one thing very surprised to us. The second approach is that we do not require normality. We simply do moment matching. That's called method of moment. Basically, we try to use the fact that the covariance of this t is given by this and the expectation is 0. So we can do this kind of moment matching by minimize this criteria. That's called moment method. And then we take derivative with respect to different variance components. It will give us this kind of estimating equation where theta are corresponding to those kind of variance components. And with the estimated variance components, we can plug into these estimates to estimate the proportion of variance that can be explained by genetically regulated expression. And let's see some results, and you know what I'm trying to say. The first simulation is that here the different columns represent different sample size from the GTEx data. We just do the simulation, and this remote zero and moment zero means that the imputation uncertainty has not been considered. So you can see that this thing is not good enough. It's totally biased. And well, after we take into the uncertainty into consideration, that's for example the remote white box, and you can see it matches well to the underlying true values. That's one thing. We just try to verify our model is implemented in a good way. Well, something surprise to us is like this. Here, we assume this beta to be random effects which follow standard normal distribution, and we also assumed alpha g follow normal distribution. However, in practice, we don't know whether this is true or not. So, for example, whether this beta, if it is sparse, that means a lot of zero, but just a few of them are non-zero. What will happen? And the same thing is that for alpha g part. So suppose this alpha is a sparse vector. So what happened to what if the alpha is sparse? What will happen to our model? And very surprisingly, that our modeling result will not be affected by this kind of sparsity. This is for the beta part. For different sparsity, the proposed method works extremely well. And for different sparsity for alpha, it is also the same case. That's a very interesting observation. And then we applied our method to real data sets. The first real data set is NFBC 1966 data. It's Finland cohort. And for the first columns is a phenotype from this GWAS data set. For example, in the middle, this is LDL, low density lipids. And for each row is one tissue. Basically, we are trying to estimate the proportion of the variance of the phenotype can be explained by the genetically regulated expression in some tissue. So, for example, here we observed some very interesting phenomena. Some tissue-specific pattern. For example, for LDL, we observed that in liver, a lot of Greg's effects have been observed there. That means a substantial part of LDL variance can be explained by Greg's. And this is the result obtained by remote estimate. And this is the result given by methods of moment. We can see the works roughly the same, except that the standard error for methods of moment is a little bit larger. So this suggests that the remote method is more statistically efficient. And here I'm trying to show you the impact. What if we do not take the uncertainty of the imputation into account? Actually, the x-axis is the good method, and the y-axis is the result from the bad method. We can see the slope is roughly 0.5. That means half of the information will be lost if the uncertainty is not taken into account. This is partly for remote, this part is for methods of moment. And more importantly, we can generalize our method simply by using the summary statistics without accessing the individual level data. Here is the key observation. Why we can do this? Because of the methods of moments, and this part only involves genotypes. And we can use some reference genome to compute all those entries in the matrix. This is simply a 3 by 3 matrix. And here it only involves the T transpose K, T, what is K? K is something like this, right? And for example, K is like this, X, T, X transpose. And if we combine together, that is, we only need X transpose T. That's simply the marginal information given by GWAS data. So actually, we don't need the individual level data. That's why we can develop summary statistic-based analysis. And we compare summary statistic-based analysis with methods of moments, which uses individual level data. And it turns out the results are quite consistent with each other. And then we apply this method to WTCCC data sets. There are 7 different data sets, different phenotypes. Something very interesting is that, for example, for rheumatoid arthritis and type 1 diabetes, if we exclude the MHC region in chromosome 6, which is known for immune system, and then if we exclude, we see, okay, the Grex effect is a little bit small. Well, if we include that part, then the Grex effect is very strong. And for those kind of autoimmune disease, but for other phenotypes, then it remains the same. That is, the MHC region does not play an important role. That means what? That means we observed Grex can contribute a lot in the immune system part, but not something else. And then we apply our method to the data, human plasma proteins. This is a new paper just published in June or August this year. And there are more than 3,000 proteins, and we take about 300 proteins because those proteins has significant non-zero heritability, so highlighted in this part. And then we apply our method to those proteins and calculate whether the Grex effect is significant. This is the QQ plot, and we do observe some significant effects in this protein data. And we ask, where are the sorry for this fig. Actually, what I'm trying to show you, where are those proteins, which protein abundance can be greatly explained by those Grex effects. And well, it turns out this is those proteins. Actually, I highlight about 9 proteins. And we show, okay, those protein abundance are highly, the proportion of the variance of the protein balance can be highly attributed to Grex effects. And we look at the details of those proteins, it turns out again, all of them are related to immune system. And then we apply our method to about hundreds of metabolites. Very interesting result is that here, first, we observe that liver, the tissue, liver, play an important role to regulate those metabolites. Right? And then we also have something else here. And finally, we apply this kind of thing to schizophrenia dataset. Basically, we have 4 different GWAS data sets for schizophrenia. We do observe significant brain, the tissue in brain, the Grex effect is really, really significant. But finally, we apply to human height, and the left-hand side is the GWAS data published in 2014, and the right-hand side is the GWAS data from UK Biobank, and we just observe a very, very tiny proportion of the variance can be attributed to the Grex effects. And well, I asked why it is the case. I think maybe this can be explained in the following ways. The first, the height, the human height, maybe because the GTEx data collects individuals whose age was around above 40. Okay. Well, perhaps it is more important to collect some expression data for teenagers. For example, the age between 10 and 18. That perhaps is more important for us to quantify this kind of GREX effects. And let me summarize a little bit. First, we noticed that to quantify the impact of GREX, it is very important for us to take the uncertainty of the imputation step into account. The second part is that we have generalized the method to summary statistic-based analysis, and we compared these two types of analysis, roughly equivalent. And we do observe many tissue-specific signals, GREX signals, and in particular, immune-related to signals are highly related to GREX. And while we need more dynamic gene expression data to explore the regulatory effects. And this is my team at Hong Kong UST, and this is my PhD student who did this project with me. Okay, thank you for your attention.
So, yeah, thanks, Dr. Yang, for the interesting talk on the heritability and the trait. Any questions from the audience?
I was wondering if your new method might be useful in trying to identify genes that are particularly important and are regulated in different ways between humans and chimpanzees. So things that really might be essential for determining what's really human characteristics.
I haven't tried that, but for identify gene part, actually I have another paper. That's my original plan called COMM. That paper can, that method can be used for identify some specific gene through regulation effects. Okay, thank you.
Any other question? So if not, I have one question. So about the model you use, if I understand correctly, you mean you want simplicity, therefore you use a linear model.
Yes.
Right. So do you think nonlinear model may be more appropriate? And if so, what is the challenge of introducing nonlinearity?
Okay. I think for this human genetics study, most of the model, actually we have tried linear and nonlinear parts, but we have to say Fisher is really great. Actually, this model, his linear model, it turns out to be always to be the stable and the robust one, and the result can be more reproducible based on this kind of linear effects. The reason is that because this beta, the effects for each genetic mutation is really, really small. It's really hard to improve this kind of thing by add some nonlinear parts, because all those kind of signals are really, really weak. In presence of this kind of weak signal, we prefer linear structure. It's more stable and reproducible. Okay, thank you.
Thanks, Dr. Yang again. So we have a...
Okay, so I have actually a very political question. I know that this kind of study has lots of competitors from population genetics and statisticians. It's very competitive. How can you survive this competitive?
How can we survive?
Survive, yeah.
I think we need more collaborations. Yeah, and also I think one thing very important is like we need to really look at real data rather than just play around with the simulation, and make sense of the real data. It perhaps is most important thing. Thank you. Right.
Okay, great. So we had a break, and let's come back.